Well, good morning, everyone. As I quickly introduced myself earlier, my name is Anna. I'm a PhD student studying with Mark Villand, working at the forested Cabo sites in Quebec. Today, I'm going to be presenting some of my work examining the ability of remotely sensed plant spectra and how it could act as an indicator for different biodiversity dimensions, so the taxonomic and functional level. So as Etienne already mentioned, kind of the interest in remote sensing of biodiversity really stems from the fact that we can quantify biodiversity across different dimensions quite well in the field. However, from the map shows, like it's very limited in space, but also in time. And so having continuous data products could really help fill in the sparseness, which is important as humans can increasingly put pressure on our ecosystems, but also as our societal values change and we start our valuing ecosystems in a different way than we used to. So one approach for using hyperspectral products is using the information contained solely in the spectra and seeing how well it could act as an indicator, a proxy with other dimensions. So we know at the leaf level, plant spectra are indicators of unique biological components within the community. It reflects the traits that plants have and that different plant species have varying traits. So there's been quite, this relationship at the leaf level has been demonstrated that the spectral dimension of biodiversity positively relates to the taxonomic and functional dimension. However, there's a series of potentially confounding variables at the remote sensing scale. And so this approach of only using the spectra solely to quantify these dimensions that we usually measure on the ground, we're kind of less certain of the relationship. So I will be examining, or have been examining, the ability of plant spectral composition and diversity as an indicator of taxonomic and functional composition at two parks in southern Quebec, Parc National de Montmagnetic and Parc National de Mont-Saint-Bruno. Taken together, these sites span a climatic gradient across which the northern temperate forest transitions to the boreal temperate forest. To quantify the taxonomic dimension, meaning the identity and the abundance of individual species, we systematically established forest inventory plots across the climatic gradient. In these plots, we identified all the trees that met our selection criteria to species and took a suite of additional measurements associated with canopy size, orientation, and the vertical position. Using these field measurements, we then modeled the tree canopies and calculated the relative abundance of what we're calling the visible canopy area. So we think this metric would better capture what the sensors are seeing from above. For the functional dimension, leaves were harvested from individual plants at the two sites, and a variety of leaf traits, both structural and chemical, were determined in the lab. By weighting these trait values by our abundances from the forest inventory plot, we then have a community weighted mean, so the average mean trait at each of our on-the-ground sites. The spectral dimension encapsulates the spectral reflectance across the different wavelength bands sensed by the CASI and the SASI sensors. We extracted the spectral points from the directly georeferenced hyperspectral point clouds that the McGill lab produced. And then these spectra were further post-processed, and Christine is going to talk a little bit more about the stages of additional post-processing we did. And then one thing to note here is because the SASI and the CASI sensors weren't co-aligned, I did my analyses twice. So the spectral dimension is either described by the CASI information or the SASI information. So my first objective was to examine the degree of correspondence between the spectral, taxonomic, and functional composition. So this is taking a multivariate approach where the variation in the different biodiversity dimensions, it hasn't been summarized into a single metric yet. To do this, I first coordinated the data to reduce the dimensionality, and then conducted a Procrustes transformation. And this is when the different ordinations are rotated and scaled to try and minimize the sum of squared differences between the orientations. And then we can test the significance via permutation. So really this example, this framework gives us this test statistic, which is associated with the degree the two matrices are co-aligned or are associated. So we found that the Cassie spectral composition better corresponded with both the taxonomic and the functional dimensions than the SASSI spectral composition. And then kind of, as we didn't expect, we expected to see a more obvious trend because ultimately it's the traits driving the spectra and not species identity. But we only saw a marginal difference between the correspondence between the spectral data and then the taxonomic and the functional. And to kind of get an idea of how related these different values are, like 0.4 at the highest for the fit, if we compare it to the same approach, the Procrustes transformation of the taxonomic and the functional data matrices, which we know are highly related because the taxonomic information was used to calculate the functional, we can see that like overall the correspondence between the spectral data is relatively weak. So there's a big decrease. My next objective was then to summarize this composition, so summarize the variation within composition into single diversity indices, and then examine how related the different indices were. So first we calculated very commonly used diversity indices for each framework. I don't want to get too into the definitions on the spectral diversity indices, but two of them, the coefficient of variation, and then the indices Etienne and Anna Schweiger proposed that based on spectral variance, used the reflectance values at each bandwidth as input, whereas the convex Hall volume was really based on like how each plot was defined in the spectral space. So once these were calculated, we just looked at simple correlations between each of the indices. Not surprising, since the Cassie composition had higher correspondence, we see the same thing with the SASSI, or we see the same thing here with the diversity indices, where the Cassie spectral diversity indices were more highly correlated from the SASSI ones. So from here on out, I'm just going to focus on the results from the Cassie data. So we found that the spectral diversity indices were more related to abundance weighted taxonomic indices. So we can see S here is just richness. So that's the number of species present in each plot. And it doesn't include the different abundances in which they are. And you can see it's a lower correlation than the other three diversity indices. And we also conclude that species evenness. So an even community is a community that doesn't have like one very abundant species. Like every species present is occurs at a very similar abundance. So we can see that species evenness is more strongly related to spectral diversity than species richness. For the functional indices, I'm looking into kind of the math behind functional evenness and functional divergence to try and figure out why these ones were like so uncorrelated. So more information to come in the future. But we can see that there's only a slight increase in relatedness between the taxonomic indices and the functional indices that are highly correlated. So there's a little bit more information in the functional component that is getting picked up by the spectral diversity measurements. However, overall, there's weak to moderate correlation between these different diversity dimensions. And as I just mentioned, kind of against intuition, we only saw a slight increase between the functional indices and the taxonomic indices. So next, I just want to further examine the relationship between the most correlated indices, and for this talk, I'm going to be focused on the Scheinman-Weiner index for the taxonomic indices and the functional dispersion for the functional indices. And when you just fit a simple linear model to it, we can see it's not doing that bad of a job, like 20 to, 24 for the taxonomic and 35% of the variation on the ground diversity indices are being captured by the spectral diversity indices. However, we kind of noticed that these weren't what would better fit versus a linear relationship is, it is this like curve suggesting that taxonomic and functional diversity increase with spectral diversity, but only to a point, and then beyond which increases in spectral diversity aren't related to increases on the ground data. So some takeaways, we found this direct approach of applying only the spectra was found to have weak to moderate performance if you want to know what was on the ground. In general, the CASI sensor performed better than the SASI, and that there was only like slight differences with the functional diversity being marginally better or marginally more related than the taxonomic diversities. So the question of what is driving this weak to moderate correspondence, it potentially could be error at any, like a measurement error at any of the different dimensions, and it's not mutually exclusive. So we can't separate, like, if there was a way to exactly figure out the magnitude, it really probably is a combination of different errors that are introduced with the measurements at every stage. And we'll continue to explore, trying to quantify or at least estimate the different potential sources of error. One that we have looked into a bit is we have some evidence of intraspecific variation, which wasn't captured in my functional composition or diversity because we use those community weighted mean values. So here I'm showing just two species that have a broad range across the climatic gradient of the forest sites. So paper birch and balsam fir, and then just one trait, which is total nitrogen. And we can see that despite the small sample size, there is a change in nitrogen levels as you go along the climatic gradient. Here expressed from like low elevation to high elevation. And we can see that this trend is mirrored in the remotely sensed spectra. So from the field-based data, this is spectral points that fall within those modeled tree canopies of each species. And on the Y here, the first principal component of the spectral data. So we can see there's also, an increase in values that across the trend. So this is something that I'm going to continue looking into. So as I said, I'm going to try to estimate the magnitude of the different sources of error. And then really the other next step would be using a different approach to estimating biodiversity. So Christine is going to present a little bit today on adding additional explanatory factors, where it's no longer just spectra solely, but also topographical indices. I really like to acknowledge the many members of CABO, the leaf trait data from the UDM team, and of course, the spectral data from the McGill team. So many thanks, and any questions are welcome.